Okay, thank you. It's a pleasure to be here. Should we turn down the lights a little bit, or is it okay? Can you see the slides? Okay, so this is we have to do with this. Okay. Well, it's a pleasure to be here. I think this is a very exciting meeting and an exciting group of people. So I hope I have some chance to interact with you during the day. I'm sorry I haven't been able to participate so much in this meeting because there's been a parallel meeting, a European meeting of systematic biologists, and I've been drawn into various activities over there. So, but today I'm here at StanCon. Okay, so I'm going to talk about Bayesian phylogenetics and our work towards more flexible modeling and inference solutions for problems in that domain. And I just assume that a lot of you are not biologists, so I thought it would be good to start with some basic biology. Well, first perhaps we should ask this question: what is Bayesian phylogenetics? So simple or quick answer is that it's a Bayesian analysis of problems involving evolutionary trees or phylogenies. So we need to understand these trees and how we model them. And to do that, some basic biology. So if we look at a cell and how a cell reproduces, it looks something like this, right? So a cell has the genetic material, the DNA, packaged into things we call chromosomes, so the blue and green here. And before it reproduces, it replicates that DNA, so it creates copies. So at the end of that process, we have 2 copies of each of the chromosomes in the cell. And then we have this cell division process where we form 2 daughter cells, and those 2 copies get separated into each of those daughter cells. So at the end of this process, we have 2 daughter cells that are identical to the mother cell. Well, actually, they're not quite identical because during this process, of course, there are small errors or mutations, changes in the DNA. So the DNA in those 2 daughter cells will be almost identical, but have small changes. There will be some probability of some changes occurring during that process. So now you can imagine we can represent this as a tree in a tree diagram, which we call a phylogenetic tree or a phylogeny. Where the nodes represent the cells just before they divide into daughter cells, and they represent the DNA replication, if you wish. And then the lines or the branches in this diagram, they represent what happens between those cell division events, where there can be some changes in the DNA in the form of mutations or substitutions. And to build a stochastic model, what we need is some model of the branching process itself, and then some model of the substitution process, so how the DNA sequences change during this process. And maybe as a side note, I should say that the biologists always, well, these are trees, right? So we draw them with the root down. I've noticed that a lot of computer scientists tend to turn them upside down, but for a biologist, this is how we like to draw them. So, of course, in biology, it's much more complicated. There's always complications, and in this case, in particular, it's a sexual reproduction that creates complications. So, in many organisms, we have 2 different kinds of individuals. So we can call them plus, minus, or we can call them females and males. And those individuals, when they produce progeny, they combine their genetic material. So we get these networks of genetic connections between individuals. And over time, you can think of this as forming of these interbreeding individuals, forming something that we call a population or a lineage or a species, an entity that's separate from other such entities. And if you look over a longer time scale, we tend to see in evolution that those lineages get separated into sublineages. by barriers, and if those barriers persist for a sufficiently long time, then these lineages develop differences such that they cannot interbreed when they meet again. And then we call that speciation, and you can think of that as being, we can represent that in a similar way with a phylogeny, where the nodes now correspond to these splits, the speciation events. Okay, so there's lots of applications of Bayesian phylogenetics, or these types of problems occur across the life sciences. I just listed some examples here to give you a feeling for where these problems occur. So in medicine, we have problems like virus transmission pathways, epidemiological models, predicting next year's influenza outbreak, identifying pathogens. All of those so typically involve evolutionary trees. In classical biology, we're using these methods to study relationships among organisms. That's underlying how we classify them. Divergence time estimation is also based on this. In molecular biology, we're using them to study evolutionary processes, selection analysis, which genes cause some phenotypic trait X. In macroevolution, we're using them to study patterns of diversification and extinction over long periods of time. Biogeography, where does an organism come from? And the list just goes on. And it's one of the reasons why the software doing this, it gets widely used. Okay, so just to give you a feeling for the nature of these models, let's consider the models we use for trees, or the branching process, and for the substitutions. So for the branching process, it's so one very popular choice is these classical birth-death models with a birth rate and a death rate, and then we condition them on some time of origin or a time of first speciation, so that the first speciation among a set of extant lineages, we call that the most recent common ancestor. So, but typically with that combination, we can generate a prior inducible probability distribution on the shape of the tree, or the topology, and the speciation and extinction times in that tree. So together they define the tree and the branch lengths in that tree. Often in these trees, if you have a death rate, of course, you have some lineages that go extinct, and often we cannot observe those extinct lineages. So that's one important problem when we have to integrate over all the possible ways in which there could be extinct lineages that are unobserved, these problems. Okay, so to construct or discuss the substitution model, we need to know a little bit more about the DNA sequence itself. You can really think of this as a sequence from a 4-letter alphabet, consisting of these 4 letters, the nucleotides adenine, thymine, guanine, and cytosine. In the DNA molecule, we really have 2 DNA sequences, a sense strand and an anti-sense strand or sequence, and they pair together, and that's important in the replication of DNA. But you just think of this as the DNA sequence as being something written in a 4-letter alphabet. And then the substitution model. So typically we assume that the sites in the DNA sequence, they evolve independently of each other. So that simplifies computations considerably. It's also standard to assume that when the states at the tips, we can observe them without error, so that the observations correspond exactly to the states at the tips of the tree. So then the unobserved ancestral states in the tree become something that we need to worry about in our modeling and in inference. And the standard approach to modeling this substitution process along the tree is this discrete state, continuous time Markov chain model over 4 nucleotide states, and I think many of you are familiar with these types of models. So common choice here is the most general of these instantaneous rate matrices that are they're time reversible. And such an instantaneous rate matrix, you can factor it into 2 components. So the stationary state frequencies or equilibrium frequencies, and exchangeability rate. So the intensity of exchange between pairs of states. So this is common structure of a phylogenetic or evolutionary model. So there's some specific challenges in this domain. So one of them is that tree space is huge. So you don't need to increase the number of tips very much before you have an enormous number of possible topologies. And if you want to sample over them, of course, that's going to be a problem. And then we have this mix of the discrete topology, shape parameter, and a lot of continuous parameters evolving on this. Or being dependent on these discrete variables. So that's also a challenge. And then the complexity grows quickly with more realistic evolutionary models. Perhaps you have to drop the assumption of site independence, etc., etc. So it quickly becomes quite challenging. So just to give you a brief history of Bayesian phylogenetics, so Bayesian approaches and Markov chain Monte Carlo algorithms, they were introduced. They were not introduced into this field until the mid to late 1990s. And one of the first pieces of software that actually was usable and could do this was Mr. Bayes, and I was fortunate enough to get, for some random reason, got drawn into the development of Mr. Bayes right from the start. So I've been working in this field for 25 years now. So MCMC actually was hugely successful. This is the first time actually that we could do parametric statistical inference for these types of problems for any realistic problem sizes. So there's a period when people were considering more and more complex models and the MCMC and using the MCMC machinery to address them. Eventually this led to there was software like Mr. Bayes really couldn't handle all models, of course, so people were writing their own software to deal with their favorite model. So we had we still have lots of dedicated software for dealing with particular types of models. So there's been a need to develop more generic approaches, generic software that can address a larger model space. And that's been a major focus in my work over these 25 years, trying to address that. In the last decade or so, we've also been flooded with data from these new genome sequencing or massively parallel sequencing platforms. So so what we've seen over that period is more and more the Bayesian approach has been suffering from these scalability issues. So there's been a resurgence of simpler models and fast and approximate maximum likelihood methods, and it's also something I haven't worked that much with that, but that's something I'm hoping to devote some time to over the coming years. So it's just to quickly introduce you to Mr. Bayes and the way we try to cover model space in Mr. Bayes. So it's really a set. This is a Mr. Bayes script that a user would in which a user would specify the model that they're interested in analyzing, and really a set of keywords and settings, keywords, values, pairs, right? And then the challenge for us in the Mr. Bayes backend, the software itself, is to figure out what the user wants from these settings. So you can construct these complex schemas and try to guide the user. This is how you flip the switches with these keyword value pairs to get the model that you're interested in analyzing. And this is not a very flexible framework, and so it's obvious fairly soon that we needed to do something a little bit more generic than that. So so around 10 years ago, we developed RevBase, which provides, it's based on probabilistic graphical models. We're trying to apply them to phylogenetic models, and we provide them in uninterpreted language, so you can interactively build these graphical models for phylogenetics. So that's the basic idea in RevBase. And I think you're, so I've learned graphical models from the documentation of bugs. So I guess this would be familiar to most of you here. So in a graphical model, you typically have at the uppermost layer, you have a set of constant nodes, right? So fixed parameters of some priors. You have stochastic nodes and the relations between them, and at some point you clamp these stochastic variables, random variables in the model to some observations. And typically also you have these plates with replication or IID. You represent IID distributions or variables. So now we can try to apply this conceptual framework to phylogenetic models. So for instance, the instantaneous rate matrix Q, you can think of that as being a deterministic node in this terminology, and it's really composed of these two random variables, the stationary state frequencies and these exchangeability rates. And then if you're thinking about what happens on one branch in the phylogenetic tree, we have an ancestral state denoted here Sij prime, and it evolves into the state of a descendant Sij, and that probability distribution is determined by the instantaneous rate matrix, the ancestral state, of course, and then the branch length, which is denoted V here. And then this is a plate is replicated over the sites in the sequence. So using this type of approach, it's easy to represent a phylogenetic model on a fixed tree. So it all fits these classical graphical model concepts. But what do we do when we want to sample over phylogenetic trees? So then the graph structure is a random variable itself. So we came up with this way. We call it a tree plate, but it's really a way of illustrating how we piece together a tree from a root. We have some assumptions on what happens at the root of the tree, and some assumptions about the internal branches in the tree, some assumptions about what happens at the tips. It's a way of illustrating this part of the model, but really we can't really use this in the software to allow the user to influence how this is structured. So if you look at sort of the overarching approach in RevBase, you can think of it as being a modular representation of the model, where this core box, the phyloCTMC object, it's really something that's dealt with in the back end, so it's entirely back end, and the user can't really influence that. But then we have these parameters feeding into that model, and those, as long as they can be represented as graphical models, we can handle them with reasonably efficient MCMC machinery in RevBase. That gives the user complete control over these modules that are outside of the phyloCTMC object. So in the last 5 years or so, we've tried to take this one step further and work with universal probabilistic programming solutions. And this has evolved into the Tree People project. And for each of these steps, I guess I've been involved in larger and larger collaborations, and in this the Tree People project in particular is definitely a collaboration between computer scientists and computational biologists. In particular, we've had some statisticians involved as well, but it's really, we're trying to create something something that's more than each one of us could create on our own. So now we get into things that are very similar to what you do in Stan. I'm not an expert in Stan. I'm trying to understand exactly how Stan works, but there's definitely similarities and I think some important differences between what we're doing and what you're doing in Stan. So I guess the vision is fairly common between us. So we want to tell the user that you describe the model, focus on that, and then you get the inference for free, more or less. And in practice, I think maybe it differs a little bit because we ask the user to write a computer program, and really like a classical computer program, the way that a user specifies the model in Tree People. And the computer program, you can think of it as a program that simulates from the model, and it clamps or it associates some random variables with observed values. So it generates weighted draws, if you wish, from the model condition or the target distribution, if you wish. And then, of course, the task for us is to make sure that from such a program description of a model, we can generate automated inference machinery that can learn the values of the unobserved variables in that model. So a probabilistic programming language, or PPL, or we pronounce it people because it goes well with Tree People. It's really any programming language, but we do require two special types of constructs. So one is this assume statement, which defines a random variable and the distribution is drawn from, and then the observe statements that associates random variables with a distribution and an observed value. So it's an observed value and associated with the distribution, and it weights the simulation with the likelihood of that observation. This is a coin flipping example, just to give you a feeling of how this works in practice. So we have what we call a model function, which is a little bit like the main function in a C program. Typically for flexibility, we wanted to take the data as input. And in that program, we define a suitable prior for the parameter, in this case, the probability P of obtaining heads. Then in this case, we have a loop describing how the observations are drawn from this, the appropriate distribution. And then at the end of this program, we return the parameter or the parameters that we're interested in doing inference on. So the user really defines what we want to get out of this program. Just to describe this in terms of expressiveness, so the tools, so Mr. Bayes, I tried to describe to you, is based on parameter settings, right? The inference algorithm is part of the software tool. The probabilistic model is also part of the software tool. You have these switches that you can use to influence exactly which model you're using. So in RevBase, I tried to describe how we give the user complete flexibility over parts of the model, but parts of the model we still need to implement in the back end in RevBase. And in universal PPLs, really the idea is to completely separate the description of the model as an independent program, and then the inference algorithm is part of the software tool, so that's a separate thing, completely separate thing. So so models requiring universality and as opposed to the standard graphical models, these are models where the graph structure is a random variable, or the number of random variables is a random variable itself. And there's lots of phylogenetic samples of this, of course, if you want to sample over tree space, if you want to sample over change history, so what happens along the branches. So there's some complex substitution models that really require us to do that because we can't integrate out those change histories. There's some examples there of that. And these diversification or speciation extinction models where we have, I alluded to that before, it's an infinite space of unobserved side lineages that we have to deal with. And I just illustrate that here. So we have one and the same observed tree with the tips that survive until today. And then we have to consider all the possible extinct side lineages that could be associated with that tree. We just don't have observations of them. So, so definitely, it's been a lot more challenging than I initially thought to try to develop these universal PPL solutions. There's a cost of generality, so we can deal with any program. We know how to do that, but what we don't know is how to always generate efficient inference. So we often end up with quite inefficient inference for those models. How you write a program can affect the efficiency, the inference efficiency. So that's not very elegant. So you'd like to get away from that. You can ask the user to provide some inference hints, and because a probabilistic programming language is, it's a powerful programming. It's a Turing-complete programming language, right? So you can do a lot, but then you have to also know a lot what happens after that in the inference step. Then the libraries of predefined model components is limited, I guess, in most of these universal PPL languages. So in the Tree People project, we try to address these challenges specifically for phylogenetic models. And the focus is on Monte Carlo samplers, so SMC algorithms, MCMC algorithms, and mixes of those. So the Tree People compiler architecture, it has some similarities with the Stan infrastructure. I think we're basing the compiler on OCaml, which is interesting, I think. And the compilation happens in two layers, if you wish. So there's a front-end compilation where the Tree People program gets converted to, you can call it an intermediate representation, a core people program. And then we combine that with inference runtimes and generate an executable. And the executable, I guess this corresponds to what you would call in Stan a fit object, I think. And then for the user, they interact with this through a Python or R interface. That's what we're trying to make sure we have in place. So the user needs to write a model script in the Tree People programming language. And then input the data, run inference, and analyze results. We want that to be done in Python or R. The Tree People language, it's a simple functional language. It's inspired by OCaml, since we have OCaml infrastructure in the back end. It makes sense. We wanted to simplify the implementation of inference algorithms, so the functional, just keeping it functional, this just helps in that respect. We do provide some syntactic sugar support, some common imperative constructs that we think would be helpful for users. And just want to give you a feeling for how this, how we manage to generate inference machinery for these programs. So basically, you think of you run a probabilistic program, and if you collect it, if you store it like a stack trace, so that's the blue line here. You can in that stack trace, you will have some assume statements, you have some observe statements, right? And then in the end, you get some parameter values, and you get the weight for those parameter value that draw from the target distribution. So to implement an MCMC algorithm, what we do is we go into that stack trace, we select one or more assumes, and then we modify the value that we have currently for that assume or those assumes. And then what we need to do is compute the weight of the that simulation with the updated value of the assume. And the challenge here, of course, is that if you change the value in the middle of a stack trace like that, you can alter there can be some consequences for the downstream execution. So really, to be on the safe side, you really need to re-execute the program from that point on. Anyway, at the end of the day, you have those two different values, and you do accept and reject, like you do in MCMC. In the SMC algorithm, instead, what we do is we run the program up to the first observe. And at that point, we have some partial executions, and we have some weights for those. Then we can do the resample required by SMC. And once we have that, we extend the program or with a proposal. And eventually, we end up with a complete sample from the model, or set of complete samples from the model and weights from those. So the challenge with these relatively simple approaches to generating inference machinery is that MCMC on large models requires really efficient local updates. So we don't want to rerun everything downstream, a place where we change something. I really want to just do the minimal number of recomputations needed to compute the weight of the new sample. And in SMC, the way you write the model program, if you use this naive approach, is really affecting the efficiency of inference. So to address those problems, we're exploring concepts from functional reactive programming. So the idea is to, from the probabilistic program, we generate a graph generating program, I guess, at compile time. And at runtime, what we're doing is we have this interplay between a dynamic graph that knows how to reconstruct itself. So it's like a graphical model, but an instance of a graphical model. And then the proposal can modify parts of that graph, and the graph knows how to update itself to create another instance of a graphical model. And then we can do the accept, reject, and the dynamic graph object, it knows how to redraw itself in response to what happens in the accept reject step. And then we started working on a layer where the user can compose inference by affecting the proposals in this type of context. So this is just a simple example. This is a geometric distribution where we have recursions, and when we stop that recursion, we return the value that we draw from the geometric distribution. And you can represent this program with just a graph that represents an instance, one computation through that program. In this case, we have two iterations, two recursions, I should say, in this particular instance. And now this graph knows how to change the number of recursions when we modify random variables in it. So these graphs, these are just two examples. This is a graph generated for a tree inference problem to the left, and a larger birth-death model. So these graphs quickly become complicated, but here the essential thing is that we can modify parts of those graphs, and we know which parts of the graph that we need to update. The R interface, I mean, this is similar, I guess, in many ways to what you provide in Stan. So I'm just gonna skip over that. If you're interested in seeing what we do, we do have a website. I mean, Stan is so incredibly mature and sophisticated compared to what we're doing. We're still at an experimental stage, I would say. But there is some information there on how to get started, installation, running your first model. There's some documentation there. And we have a repository with some examples of model scripts. And I think I'll end by giving you just a few examples of the types of models we work with currently using tree people. So perhaps most of the work we've done is on these diversification models, where we allow birth and death rates to vary along the tree. So the different lineages have different speciation and extinction rates. And so one of the models we've looked at is the cladogenetic diversification shift model, CLADS. It was presented in 2019, and this is a typical story of a model like this. You know, a good paper in a good journal, and there's some software that implements that model. Then it doesn't get maintained. Odile, who was the first author in this paper, gets she goes to industry, and now there's no really stable software that can implement that model. So three people, the kind of approach that three people and Stan, I guess, represents. I mean, this is just trying to get community behind a more generic solution. This is just really important for us across science, I guess. But anyway, this is a lineage-specific diversification model. The speciation extinction rates change at speciation events. There's a stochastic effect, which is modeled by a log normal distribution, and there's a time trend, which you can think of that as accounting for density dependence. The more lineages you get, the slower the rate of diversification. That's a typical pattern we observe across organisms. So to represent that model, is I just want to give you a feeling for the structure. So we have this model function, the top or main function in the model, and it draws some parameters from priors, and then it returns whatever we're interested in this case just the speciation rate, but we can return anything we like from that model, from the sample from that model. But then this top function, it calls other functions. So we have an observed tree function that simulates the process on the observed tree, and then we have a survives function that accounts for, simulates the extinct or unsampled side lineages, and we know they have to go extinct because we have not observed them, right? So there are some conditions we can use there. And then, of course, this is the beauty of tree people and of Stan as well, of course, that with minor modifications, you could describe any diversification model you like, and the inference machinery hopefully will deal properly with that. So now we're at the stage with this model, at least, that we can have this magic generation of state-of-the-art inference algorithms, so MCMC and SMC algorithms. We're also looking at tree inference, where we found a way to write the tree inference problem so we can generate this SMC, the SMC algorithms that have been presented or discussed for tree inference, and study their behavior. And again, these are algorithms that are not widely available. So again, the importance of having generic solutions that allow us to experiment with these inference algorithms. Okay, and this allows us to compare performance with MCMC and develop mixed SMC-MCMC strategies. So this is not as well developed yet. And then the final example is these host-parasite models, where we have two trees, a host tree and a parasite tree, and they interact with each other, and we have to account for that. So these are quite complex models, and it's fun but challenging to work with them and try to develop inference machinery for them. So in this case, the problem is that we really need to sample over a large parameter space. We can describe efficient proposal mechanism, and we can do that in the program itself. So that's what we're exploring now. So the current status, if you're interested in what we're doing, you're welcome to play with it and interact with us. There's the documentation and interface are still work in progress. We're focusing now on the graph representation, which is still an experimental. I would say they're still experimental. Inference efficiency, we have a lot of work to do there. And the interface for composing inference algorithms, and then expanding model libraries and inference strategies. We have some of that in place, but there's more work to do there. And the current model focus is basically what I've described. We have been using tree people for some simple hierarchical models as well. I guess we could have used Stan instead, but it's an important step for us in showing that the basic stuff works in tree people. And we're planning a first user workshop later this fall. And before I finish, I'd just like to address the first question I will get. So why the heck are we we're developing this separate tool instead of using or trying to expand the capability of Stan? And I think, there's been some work actually in getting STAN to work for phylogenetic models. So this is one example I know of for Mont and Darling in 2019. So they presented PhyloStan. So at that point, at least, they were still working with fixed trees, but they can then STAN can do really amazing things with fixed trees. They've been experimenting then with external loops that samples over trees and then asks STAN to do inference on each of those trees. So that's it for me.
Thank you very much. Very interesting talk. Impressive work. So any questions? We have quite a bit of time for them. So I think the first one was there. Wow. Who has the mic? Yeah. So the first one down there.
Thanks for a very interesting talk. It is very interesting to see, like, I think that chart that described how the model gets built into an executable and then plugged into Python and R, I could just like kind of white out and put different labels on it and it would look exactly like the STAN pipeline we have in like command STAN R, for example. A question about the tree people language. These assume observe statements, can they happen in any functions or are they restricted to that? Like, what is special about that model function compared to other functions?
If you're thinking of a model description or a model program or a script, we still struggle with the terminology there. But so it can consist of several different functions. One needs to be the model function, but the assumes and observes, they can be live anywhere in the program. So we often place them in other functions called from the top function or the model function. So there's nothing special. Did that answer your question?
Yeah. It's just that sounds like it solves a problem we always have in STAN, which is that it's hard to write, for example, like a hierarchical prior as like a reusable component in STAN, because it would have to touch multiple, like STAN, you have to put all the parameters in one place. But if you can put these observe and assume statements other places, you can kind of build up a model of, or a library of model pieces more easily. So that was just interesting.
Yeah, I wish we had spent more time studying what you do in STAN. I think we could have learned a lot, and maybe there's something we do that could inspire something in STAN. We'll see. But it's very interesting. I really enjoyed the workshop Monday afternoon on the STAN compiler.
Yeah, I think there was a question right behind there.
Ready? Thanks very much. It was a great talk, very inspiring. You mentioned in passing the importance of community and building a community base. So I just wondered, did you take any kind of structured approach to engaging that community to help you design the language? Was there a kind of co-creation aspect to it?
No. I think the answer is no. I do have quite a bit of experience working with the empiricists. And I do empirical research also in my lab. So I feel we have a decent understanding of the community, what the community needs, and maybe we should have been more active in engaging the community. It's the community really. I think you probably have the same thing, right? But in biology, it's very distinct. So we have these empiricists that really want something they can use, just a recipe to follow, right? And then we have these computational biologists, these people that can write a piece of software themselves. They understand the models. I guess the primary users, at least at this stage, for us is those more advanced developers and of models and inference machinery, etc. So the workshop we're organizing later this fall is the first instance where we actually get those people together in a room and a small group of people and see what if they like what we do or not. But we have had a little bit of conversation with a few of those key people, but. We've really struggled to understand how to sort of tame the beast. The universal is great because you can write any model you like. I mean, it's Turing complete. So, but how do you generate efficient inference machinery? I mean, the things like. Simple things we've done in computational phylogenetics for a long time, like raw Blackwellization. So we integrate out things, and now we need to do that automated without having the user to interact with analysis. So it's lots of challenges. We really haven't been prepared, I think, to talk to users at length yet. But we'll see how this goes.
Thank you.
I think Paul was the next one.
Okay. Thank you. Thanks for the talk. You did mention that there are cases where people build their own software, and then if they leave for industry, the software get kind of thrown away. So, do you have any plans how to kind of maintain your software for a long time? You're talking about community, but do you have any other plans to get like a more stable funding source and to kind of keep it alive for, let's say, 5-10 years?
Yeah, I've been thinking about those things, and for me, sort of approaching the end of my career, realizing that at some point I need to retire, it's really it's something we need to think seriously about. I think you have some interesting solutions in the STAN community, and I'm not quite sure yet what's going to happen. I mean, there's a lot of experimentation now with these universal PPLs all over the place, and we'll see how the concepts or the approaches that we develop, how well they stand the test of time, and whether it makes sense to perhaps merge our efforts with some of the other universal PPL efforts. Or we should try to maintain this as a separate community. If people have ideas on how to ensure this long-term maintenance, I'm all ears. But it's an important problem. It seems like in the US you have more better understanding for the need of this long-term maintenance. I think NSF has played some role in trying to support these software solutions that are widely used long-term. I have a little bit of support for MrBase. I keep it. It's not actively developed anymore, but I have a little bit of help from the bioinformatics infrastructure in Sweden to maintain it, which is great. So we are discussing those things in Sweden, and I think we'll probably hopefully see over the next few years more discussion on these long-term questions. Well, software is all over science now. It's really dependent on software.
Okay, thank you.
Okay, so I think, Paul, maybe we could take it afterwards. We can move on because there's a contributor session. We want the younger generation to get their time too, right?
Yes, absolutely.
So let's thank Fredrik once more.